Honors biology folk. Well, while some of you are taking the test, the rest of you are stuck online with Recorded Me. We're going to begin the next unit, which is transport and enzymes. It's going to be a pretty small unit, so expect another test pretty quick, just FYI, all right? We're going to do transport right now, and we'll do enzymes in person. How about that? But this transport usually goes pretty well, so it's a good way to record a lecture. Here's our to-do list. And the first part, oh good, I got that working. This part's going to be review from your Wednesday activity. If you did what I told you to do on Wednesday, this will be all very familiar stuff. This is going to be new for everybody, okay? But none of this will take long. This is usually a fairly short, pretty easy lecture. So membrane structure. Membranes are made of 3 of the macromolecules, lipids, 2 types, right? And absolutely the most abundant. Roughly, I would say between 95-99%. Proteins are going to be the other 1 to 5%, and then carbs are decorations. Okay, they exist and they're important, but there's not a lot of them. So let's talk about the 3 types and why they're important. We'll start with lipids. Okay, there's 2 types of lipids in the membrane. The phospholipids are absolutely the most abundant. Phospholipids are usually represented with this type of shape. These would be the hydrophobic tails, so they hate water. And this would be the hydrophilic, water-loving head. So you put it inside water, and it will form a bilayer with all the tails on the inside where they are protected, and all the heads on the outside where they can be with the water they love so much. Okay, so that's really my bad drawing. This is the more professional drawing, but notice they're not all that different. Okay, the blue is the water, because water is always blue. The heads love the water, so they're on the outside of the bilayer, and you have this kind of oily, hydrophobic, all the tails, the parts that hate the water, here protected in the middle. All right, bilayer, 2 layers, 1, 2. All right, the whole thing makes sense. This is the foundation of the membrane. The second type of lipid is cholesterol. Cholesterol is nonpolar, so it's here mixed up in the oily stuff. But you notice it's got a really odd shape. 3 circles in a doghouse is how I've always heard it described. 1, 2, 3, doghouse, okay? And it creates these gaps in the membrane because it doesn't fit perfectly. The gaps allow for more fluidity. All the parts can move around more easily, and the whole membrane is more flexible. Animals tend to have more cholesterol. Plants tend to have less cholesterol. Our membranes, we're soft and squishy, so our membranes are more flexible. Plant membranes tend to be harder and less flexible. Okay. All right, second most abundant is proteins. Proteins also come in 2 types. Integral. Integral are inserted, either partially inserted, like this guy, okay, or completely, fully inserted, going a whole way across, like that guy. So I don't care if it's partially in or the whole way in and all the way through. Both of these are integral proteins, okay? They have to have a non-polar region to go into the oily tails. Peripheral, now you know what peripheral vision is, right? Peripheral vision is what you see on the edges. Yeah, same idea. They exist on the edge of the membrane. So this guy and this guy are both peripheral. Peripheral are hydrophilic. They are polar the whole way. They love the water. They love being surrounded by the water. They want nothing to do with the tails. So they do not go into the membrane at all. They stay on the edge, right? 2 types, both pretty straightforward. Finally, our carbs, our decoration. They are attached to either a protein, like this case, in which case we call them a glycoprotein, glyco for carb, or they're a glycolipid, and they're attached to a lipid. Either way, they're on the edge, sticking out into the water. Now notice, I kind of like this picture, we still have an integral protein here and a peripheral protein here, okay? Integral protein here, yeah. But the carbs are only outside, and like they really are cellular decorations. The book called them cellular markers, decorations, same basic idea. That's not to say they're not important. One of the most famous and important carbohydrates that decorate your red blood cells make up blood type. So there's 4 blood types. You can be A, B, AB, and O. All right, if you are type A, what that really literally means is you have A carbohydrates on the surface of your red blood cells. If you're type B, you have B carbohydrates. If you're type AB, it's kind of like being ambidextrous. You have both. Not some type of way AB hybrid. You are A and you are B. You have A carbs and B carbs. Okay? And if you are mutant, and I'm sorry, this is a relatively recent human mutation, you can't make the carbohydrates. So if you're type O blood, you don't have these. Nope, 0 gone. This isn't really a problem. I'm not saying like you're going to die or oh no, oh, it's how horrible. They don't do much. This is not a lethal condition, right? You simply just don't have the carbohydrates. The good thing is, you, if you're type O, you're the universal donor. Because guys, these carbohydrates are signals to the immune system about whether or not you belong. If you're type A, right, your immune system is used to seeing these A carbohydrates and goes, oh, A, you belong here. Keep going. Happy, happy. If it sees any B carbohydrates, attack, kill, and destroy. Because those are foreign. Never seen them before. They do not belong. So type A people cannot receive type B blood. It don't go well. But O doesn't have any carbs. So the immune system ignores it. Type O can go into A, and the immune system goes, I don't see anything. It's like a ghost. I'm going to ignore it. Type O can go into type B, and it will be ignored. O can go into anybody. Okay? AB, by the way, is the opposite. If AB person, their immune system goes A is normal and B is normal. So AB can receive from A, it can receive from B, and of course everybody can receive from O. AB is the universal recipient. If you're type AB, you can get anybody's blood. Now notice I'm not telling you anything about positive and negative. Whole different conversation. Proteins, a little bit more complicated. Yeah, not going there. This is just A, B, and O. All right? Anyway, that was a fun aside. Now, one last thing about membrane structure. The membrane is called a fluid mosaic. It is fluid because everything is mobile. None of these parts are laid down, and they're all drifting around the cell. Think of a bathtub, okay? And take ping pong balls and dump them in the bathtub. That's the phospholipids. They're not nailed down. They're not connected to the bathtub. They're just floating on the surface. Fluid. Okay? Mosaic, because it's all different sorts of stuff. It's not just ping pong balls. It's ping pong balls and rubber ducks and floating boats and all sorts of stuff. Okay? You got your lipids, your proteins, different types of proteins, different types of lipids, the cholesterol's in here. All this mixture, mosaic mixture, floating around fluidly and loose. Fluid mosaic. All right. So that is one part done. Not bad at all. Next up, we will talk about transport. That's the next thing on our list. Transport comes in 2 types, passive and active. Okay? Very simple. Very, very, very simple difference. Passive, no energy. Active, uses energy, which is just what the words sound like. Very easy to remember. If you're passive, you're a couch potato, you sit and watch the world go around you, no energy. If you're active, you're moving, you're rock climbing, you're bicycling, you're using lots of energy. Okay? Another difference, passive is always movement from high to low. From high to low. It's like going downhill. If I'm going downhill, I'm not using any energy. Sit back and let it happen. Active moves from low to high, and I'll explain these terms in more detail in a bit. But low to high is like riding a bike uphill. You're going to work for it. Okay? You're using up a lot of energy to go from low concentration up to high concentration. We're going to talk about passive first, and we're going to finish with active. Oh, different. Nope, don't do that. Yay. All right. Diffusion. Simplest form of passive transport. Diffusion moves from high concentration to low concentration, from high to low. So we say it moves down. That is going down. Yep. Okay. So a great example of diffusion is just molecules spreading out. If I have a beaker of water and I add a drop of dye, this is a high concentration. Okay? And it spreads out to fill the whole beaker. So we start with a red drop. We end up with a pink beaker, and it spreads out to a low concentration. So high to low. Downhill. Now where'd the animation go? The animation was cute. There's the animation. High concentration spreading out to an equal low concentration. So high here in the corner, spreading out. Picture the same idea. High concentration, spreading out, moving to equal low concentrations. Diffusion stops when it hits equilibrium, when there is no more high or low, and everything is equal. Nowhere to go. Right? Same idea, but two molecules. So notice green here, purple here, high green, high purple. You can tell me what's going to happen, right? I mean, it's kind of obvious. We're going to diffuse, everything's going to move until equilibrium. 3 purple, 3 purple. 1, 2, 3, 4, 5, 6 green. 1, 2, 3, 4, 6 green. Everything is equal. Okay? So, yeah, diffusion makes sense. Biological example, breathing. I don't mean cellular respiration here. I mean breathing respiration. Okay? Guys, the air we breathe is high O2, low CO2. All right? Lungs are kind of medium, and cells are low oxygen because they keep using it up, and high CO2 because they keep making it. And we'll talk about all that process in the next unit. So, when we breathe, okay, this side over here, this is the lungs. Right? The lungs have more oxygen, which then diffuses into the bloodstream. And the lungs have lower CO2, so that's diffusing out because this is the air. Okay? The blood travels, the blood travels, the blood travels. Actually, I was supposed to say blood is medium. The lungs are filled up with air. They don't have their own concentration. Air/lungs. The blood is medium. I thought I screwed something up. Okay, so the blood's over here. The cells are low O2. So notice the O2 is moving out from medium to low. So oxygen has gone from high to medium, from medium to low. Carbon dioxide is moving in. So carbon dioxide moves from high to medium, back to the lungs, from medium to low. So the whole thing, passive diffusion. Both molecules are moving down their gradient. If this wasn't passive, if this movement required energy, we couldn't eat enough food to fuel it and survive. That's how much oxygen you need in the course of a day. So if this movement required energy to push it into the blood, to push it from the blood into the cells, guys, you could eat butter, right? Which is just total fat. And it wouldn't have enough calories to fuel this and give you enough oxygen you needed in your cells. All right, so thank God, diffusion, passive, and no energy. It happens for free. Okay? All right. One last fact about cell membranes to help with the diffusion. Our membranes are what we call selectively permeable. Selectively permeable is a simple idea. Some stuff can get in and out and some stuff can't. The cell membrane chooses what it allows to pass and what it blocks. Okay? So some things thou shalt not pass, and other things, oh, come right on in. Some general guidelines. If you are small and nonpolar, so small means that you can fit, remember our membrane here? Small means that you can fit in between the phospholipids. Nonpolar means you can get through this oily tail nonpolar area just fine. Then you're going to go across with no problem. Okay? Oxygen gas, carbon dioxide gas, the two we were just talking about, no problem. Easy peasy. If you are large and polar, yes, I know I just skipped the middle. If you're large and polar, it ain't going to happen. Either you are too big and you don't fit, okay, like glucose here. Glucose is 24 atoms. That's enough. Glucose can't cross on its own. Or if you're polar, like these ions, positive or negative, don't care, you're going to hate this hydrophobic region, and you're gonna go, I don't want to do it, and you're going to turn right back around and go out the way you came in. Okay, so large or polar, you're not crossing. Now, if you're a mix, that's ugly. Okay, yeah, I can do that. Haha. All right, if you're small but polar, so I can fit, but I got these tails to worry about. Kind of. You can cross, but it's not easy. Great example is water. Water's only 3 atoms, right? CO2 is 3 atoms. We're small. But this whole region, the tail region, the whole region the tail region exists is we hate water. And here comes water going, let me through. It actually kind of works, but not well, right? So this is kind of the in-between of yes, but slow. So yes and easy, not a chance, kind of, right? Now, for the kind of and for the not a chance, you need transport proteins. When you use transport protein, a transport protein is a protein in the membrane, okay, that somehow helps molecules get across. This is called facilitated diffusion. So since it's diffusion, active or passive? So long as I keep saying diffusion, we're still passive. So no energy here. But to facilitate means to help. So it is basically diffusion with help. The help is the protein. Okay? So here's a question. This protein, integral or peripheral? Let's review the beginning here. Integral, well, it's going into the membrane, so I hope you said integral. Okay, that was a hiccup. Moving on. All right, so facilitated diffusion, a great example is aquaporins. Aqua means water. Pore means hole. It's a water hole, literally. It is a hole, and the water goes through the hole, and that is the easier way, because water's small, but it is polar. That is an easy way to get water into the cell and out of the cell. It can go both ways. Okay. The discovery of aquaporins won a Nobel Prize almost 20 years ago. Jeez, time flies. All right, so let's see our to-do list here. Check. Diffusion, diffusion with help. Yay. Next up is really important. Really. Because it is osmosis. And your lab on Monday is osmosis. Your pre-lab is due before the end of class. Please tell me I told you that before I started giving the test out. Okay? So let's do some work and make that writing that lab report easier. Okay? So osmosis. Osmosis is very simple. It is the diffusion of water. Simple. It is passive because diffusion, and it is no energy. We are not hooking your eggs up to a battery. That would be bad. Okay? No energy required. Water moves when solutes can't. So this is, believe it or not, this is a device called a U-tube. Just not that U-tube, but a U-tube. And we have in here water solutions. We have a high concentration. Look, lots of stuff. We have a low concentration, very little stuff. Normally, the little purple balls would move, and we'd have equilibrium. But the membrane is selectively permeable, and the membrane won't let the balls move. They're stuck. So to reach equilibrium, the water moves instead, and this is where osmosis, movement of water. Okay, so the water will actually defy gravity, flow this way, so this side gets watered down, this side gets more concentrated because the waters are going away, and if you look now, we have reached equilibrium. And if you count the purple balls, the number of purple balls haven't changed. They've just gotten more concentrated and less concentrated. Right? Diffusion, the movement of water. Osmosis, the movement of water. All right, 3 very, very important vocabulary words. Hypertonic. Think hyper means more. Hyper kids have more energy. Hypertonic has more solutes, more sugar, more salt, all right, more gelatin, more everything. It's just the more. So a hypertonic solution, lots of stuff. Okay, I usually draw this. Here's the cell, and just lots and lots of stuff that the cell is floating in. Very thick liquid out here. Okay? Hypertonic. So the water, remember, water can only dilute. So the water is going to be pulled out of the cell trying to dilute all this stuff on the outside. So in a hypertonic, the water moves out. Hang on. Oh, that is, okay. So this causes the cell to shrivel up like a balloon where all the air leaves. It just shrivels. So here you see the red blood cell is shriveled up, and here the plant cell has pulled away from the cell wall. All the water has left it. Ooh, I should not draw that way. I meant to point and say, here's the red blood cell, but what I normally do is I draw and say the water has left the red blood cell. The water has left the plant cell. The blood cell is called shriveled. The plant cell is called plasmolyzed or shriveled. Okay. Now the opposite, hypo. Hypo, C-O, think 0. Hypotonic has less stuff. The perfect hypotonic is pure water, which has 0 stuff. Okay, so now beaker, cell, stuff in the cell, nothing in the water. Now the water moves to dilute. Water is going to go into the cell. Okay, so here we go. This again, for the red blood cell, this is going to be a problem. So now water is rushing in. Balloon, too much air. Boom, the cell lyses, the cell explodes. Plant cell does not explode. Why doesn't plant cell explode? Cell wall. The cell wall holds it together and protects it. In fact, this is ideal. This is the best possible environment for plants. Because plants can't drink. No mouth. So instead, they need the hypotonic to force water into the plant cells. Hypotonic is how plants drink. So this is a happy plant. It's a dead red blood cell. All right, isotonic. Iso means equal. Same inside and outside. So beaker, cell, Dots everywhere. All the same dots everywhere. Equal, isotonic. With isotonic, there's no net movement. What goes in equals what comes out. This is a happy red blood cell. It's not shriveled. It is not blowing up. This is the perfect for red blood cell. If you look, this is not a happy plant. It is a wilty plant because there's no water going in. And no water going in means problem for plant. So this is not ideal for plants, but man, oh, that's a scary looking happy face. It is a very happy red blood cell. All right. This is a popular picture. I stole this straight out of the book. So first off, we have the animal cell here, right? We have the plant cell here. Yay. Okay, let's look. We have, no, this isn't good. The water is going in, which means hypotonic. The water is coming out, which means hypertonic. If you notice, look at the background colors. Very light, very dark. Lots of stuff, not a lot of stuff. And that means here in the middle, the Goldilocks, isotonic. Isotonic is ideal for animals. Over here, turgid means stiff, by the way. This is ideal for plants. This is wilty. You'll sometimes see flaccid. Flaccid means soft. So stiff is hard, flaccid is soft. Shriveled, or we've used the word plasmolized before. This is very bad. Here, shriveled. And here, lysed. All right? All good vocabulary words, all very good important ideas to know. Definitely a huge part of your lab report. In fact, you may want to pause now and do that pre-lab that's due before the end of class. Just throwing that at you, maybe, maybe not, I don't know. But it is a suggestion before we move on. Okay, a couple of ideas just to get you thinking. Possible test questions. If you're dying of thirst at sea, okay, that's a bad idea. That's bad. But why would drinking salt water make it worse? So first off, salt water. Hyper, hypo, or iso? Hypertonic. Okay, hypertonic. All right, so here's the beaker. Here's the cell. Here's all the salt in the water. Has more salt. Hypertonic. So where's the water going to go? Straight out. So guys, you drink salt water, you get more dehydrated. You get more dehydrated. So you're dying of thirst. Well, now you're dying of thirst faster. So that when they say don't drink salt water, they mean it, because you're dead. All right, most eye drops are 0.9% saline. There actually is some salt, not a lot, in eye drops. So what do you think is the normal salt concentration of the fluids of the eye? Greater than 9%, less than 9%? Well, if it's greater than 9% in your eye, that would make the salt water hypo, and it would force the water into your eye. Well, that'd be bad. Swollen eyes, no bueno. But if the salt in your eye is less than 9%, now it's pulling the water out. Also no bueno. Remember, animal cells, including animal eyes, like it isotonic. Our eyes are 9% salt. The vitreous humor is 0.9% salt. That's why it's not 1%, it's not 0.5%. It needs to be exactly 0.9%. Then it is isotonic, and then it just lubricates without overhydrating or dehydrating. Okay? So anything that touches in your body or touches your body, best off if it is isotonic. Let's look at our to-do list. Check, 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 check. Oh, I like these two. This is the fun stuff. Finally, we're getting to active. Finally, time to use up some energy. So active moves against its gradient. It moves from low to high. So basically, go away. I'll deal with her later. Basically, we move from where the molecule isn't to where it is. And we make it more concentrated. It doesn't want to go there, so we use energy to force it there. So looking at the picture down here, we have a very high concentration of, white triangles. We have a very low concentration, right? What this protein here is doing is there you are, H+. It's taking the white triangles, pushing them to the high concentration and using ATP energy to do it. Okay? Any type of protein that does active transport is called a pump, just like pumping water. You pump water to move the water, usually to where it doesn't want to go, sometimes against the force of gravity. We pump water up to the sink. So here, our solute, it binds. The ATP gives its phosphate, which is its energy. The molecule gets pushed across, and then we get rid of the old phosphate, get ready to do it all over again. Very simple idea. The most important, the most famous active transport sodium-potassium pump takes sodium, takes ATP, shoves the sodium out, at the same time pulls the potassium in. Notice high sodium out here, and notice high potassium in here. So in both cases, we're moving to the high concentration, active transport. And it uses this energy from ATP to make it happen. So there it goes. Here it comes. Right? So, a cell actively transports calcium out of the cell. So here's my cell, and we're shoving the calcium out, using energy to do it. So this means, just this one little sentence, is calcium more concentrated inside or outside of the cell? And I'm telling you, this is active. Active is always low to high, telling me it is more concentrated outside of the cell, less concentrated inside. Make sense? Right? And then finally, our last topic, if you've studied with me for so long, I'm impressed, bulk. When you think bulk, think big. We're going to be moving large. And earlier I said glucose was big. Glucose has 24 atoms. Yep, big. Polysaccharides, proteins, thousands of atoms. Okay? Glucose takes like 1 ATP. Bulk transport takes a couple of thousand to a million. Yep. Okay. Lots of energy. Big stuff. Guys, this is 18-wheeler type transport. Okay? Huge. There's two types. I love this. The names make sense. Exo. Exo means out. So exo transport, exocytosis, we're going out of the cell. This, heck, some people are taking a test over this right now. This is where the rough ER, with the ribosomes, right, transports things to the Golgi apparatus. And then after it leaves the Golgi, it's in its little vesicle. The vesicle is going to fuse. Whoa, too big. That was a bad idea. The vesicle, so here's the proteins we've made, is going to fuse with the membrane, and all those proteins are going to go out. Exocytosis, okay? Yay. Picture. Oh, why did I draw it? Okay, same idea. Rough ER, going to Golgi. You can see the purple dots. The purple dots are the proteins. The vesicle fuses purple dots, going out. Actual, this is TEM here, electron microscopy image. This is the proteins. This was the vesicle. Okay? Looks like popping a pimple, but the proteins are in the process of leaving the cell. It's like a once in a million picture. It's gorgeous. I love it. But you can see this is exocytosis in process. Okay, the opposite of exocytosis is endocytosis. Endo is coming in. That's why I love it. It makes sense. So we're forming vesicles, and the cell takes in these huge molecules. There's going to be 3 types, depending on whether we're taking in food, liquid, or specific stuff. Okay? First off, phago. Ooh, don't do that. Okay. Phagocytosis means cell eating. We are taking in food. We are taking in solids. So phagocytosis is enjoying solid stuff. So here we have our cell. It wraps around, takes in. Oh, look, we have a food vacuole. Amoeba wraps around, and this paramecium is becoming a food vacuole. Real life phagocytosis. Oh, here's another amoeba wrapping around the bacteria. Poor bacteria. White blood cell wrapping around bacteria. Right? This is phagocytosis. Your white blood cell is eating the bacteria. Pinocytosis is cell drinking. You take in a little gulp of extracellular fluid, forms a little water vesicle, plus whatever is dissolved in the fluid. Pinocytotic vesicles tend to be much smaller, but you can see lots and lots of them being formed right here. The cell taking tiny little sips of liquid. And then finally, here's the mouthful. Oh my goodness. Receptor-mediated endocytosis. Ooh, receptor-mediated. The only, guys, all that means is you're going to pick and choose what you take in. With phago and pino, the cell just kind of goes, oh, stuff. Let me see if it's usable, and grabs it and hauls it in. It has no idea what it's taking in. Here, we use these receptors. That's the red things. The receptors are specific. They bind to one specific particle. So this cell wants purple triangles. And so it is targeting, it has these proteins seeking out purple triangles. And when they've bound to enough purple triangles, it takes in the vesicle. So it is targeted for one specific thing. And here you can see it's gathered up a lot of whatever it is and taken it in. Good example of this. This is how your liver takes up cholesterol. The liver cells have special receptors in them looking for what are called LDLs. Would you go? Whoop. Okay. These LDLs are transporting cholesterol in them. They have the proteins. The proteins bind to the receptor. The liver takes in the cholesterol. Works great. There are certain people that have a genetic disease where they don't make the receptors. Because they don't make the receptors, their liver cannot absorb the cholesterol. These LDLs stay in their bloodstream. The disease is called remember hyper means more, hypercholesterolemia. And the cholesterol accumulates in the blood. And guys, it even forms these deposits. That's little pockets of pure cholesterol that form under the skin. People with severe cases of this disease can die of heart attacks in their 20s, right? And they have to be on low cholesterol diets their entire life. Like some of your parents in their 40s, 50s might be on low cholesterol diets. These people are in high school on low cholesterol diets because their bodies can't process it, just because they don't have the receptors they need. Right? You can live with it, you just got to be careful. We survived! That's everything. Check and none, but not done. Because Sister Kelly's a mean person. Review! Quiz! I'm going to put this in Edpuzzle and hope it works. All right. So, we have a molecule. What type of transport is this? So answer and answer. No energy. That's what I see is no energy. No help. Okay? So that means diffusion. This molecule is moving completely on its own. No energy, no help. It is simply, diffusing into the cell. Ooh, here's another one. Okay, what movement is this? Let's look. There's your giveaway. Osmosis. Water. Anytime water moves, it is osmosis. All right, what type of transport is this? Let's see what's going on here. We have an amoeba. That looks kind of bacteria-ish. And it's going in, and the cell is amoeba eating. The cell is amoeba eating it. The cell is eating it. So this is bulk, specifically endo, specifically phagocytosis, because that's a solid. That is not a liquid. Oh, look, it wants to eat another one. Okay. And what type of transport is this? That's interesting. All right, ATP is energy. If that is energy, this is active. And that tells me that's a pump. Right? Not bad. Oh, one more. Hello. Okay, so looks kind of like the first one. Only we got this thing in it. This is new. But we saw a molecule just moving. I don't see any energy. No ATP anywhere. But I got this protein helper. So that helper means facilitated diffusion. Diffusion with helper protein. Okay, one more. Let's see what we're doing here. So, look, they're numbered. 1, 2, 3. So 1 is in the cell, 2 is fused with the membrane, and 3 is we are out. Exo, because we're going out. Cytosis. All right? Bulk transport happens in these vesicles, and this one is going out of there. And done. Done. Done! All right, now you've survived. Go do your pre-lab. Have an awesome day. See you later.